Welcome everyone to this month's session of the General Medical Microbiology Seminar Series, and thank you for joining us today. My name is Jose Puente, Senior Editor of the Pathogenesis, Virulence, and Host Response section of the Journal of Medical Microbiology. I am joined by my co-host, Kalain Mathie, JMN Profiles Editor, and Fionn Watson, Microbiology Society Editorial Coordinator. Just a few notes before we begin. Live transcription is available for accessibility. If you prefer to hide the captions, simply click the live transcript button and select Hide Subtitles. This session is being recorded. All participants will receive a link to the recording following the session that will be available for 7 days. If you have specific questions you would like to address during the Q&A segment, please use the dedicated Q&A function. The chat is your space. Feel free to introduce yourself and connect with fellow participants. We value your feedback. Please take a moment to complete our quick poll at the end of today's session. Your input helps shape future sessions and provides valuable insights to our speakers. It is my great pleasure to introduce our speaker, Professor James O'Gara. After completing BSc and PhD degrees in microbiology at the University of Galway, Professor O'Gara furthered his research training during two postdoctoral fellowships at the University of Texas Medical School in Houston and at Trinity College Dublin. He held academic appointments as senior lecturer in microbiology at the Royal College of Surgeons in Ireland and University College Dublin before returning to the University of Galway in 2012 as professor of infectious disease microbiology. At the University of Galway, he has served as head of microbiology and on the governing authority. In 2022, he was elected a member of the Royal Irish Academy. Professor O'Gara is here with us today to present his work on the use of purine nucleosides as adjuvants to resensitize methicillin-resistant Staphylococcus aureus to beta-lactam antibiotics. Professor O'Gara, thank you for being with us today. The floor is yours.
Great. Thank you very much, Jose, and everyone for the invitation. It's a real pleasure to be here. So what I'll do is, as Jose mentioned, I'll talk about our kind of recent and ongoing work using purine nucleosides as adjuvants to resensitize MRSA to beta-lactam antibiotics. So to begin with, I'll try and put in context why antibiotic adjuvants are useful. And then with your indulgence, I'll go back a little bit in time to some research that I've been doing with my group over maybe 20 years, which kind of led to our interest in antibiotic resistance in MRSA and looking at how that phenotype is controlled. So first of all, let me just talk about, obviously we all know about the AMR crisis and the decrease in the number of new antibiotic agents that are being produced. And so in order to overcome this, of course, we either need to produce new antibiotics, which is very costly, time-consuming and difficult to get through regulatory hurdles. But there are other approaches. This isn't an endless list. Of course, phage therapy is topical, photodynamic therapy is possible, vaccination to prevent infection, but also looking at our currently licensed antibiotics and thinking about, can we get them to work better? And in particular, this is the idea of using novel combinations of existing drugs that may have synergistic activity, or perhaps using adjuvants to enhance the activity of existing drugs, particularly against pathogens that have become resistant. And it's in this area, really, this adjuvant area, that I'll eventually come to in the talk, and how our new research is looking at purines as adjuvants to potentiate the activity of beta-lactam antibiotics against MRSA. So of course, combinations of drugs, and using drug combinations and adjuvants is very important. And finding ways to preserve the efficacy of currently licensed antimicrobial drugs is very, very important. And sometimes it's kind of overlooked. We tend to think about we need new drugs or new therapies, but kind of preserving the activity of the existing drugs or finding ways to make them better is important. And so, of course, there's a lot of interest now in repurposing currently licensed antibiotics in new combinations, which can be sometimes more complicated than it seems, but potentially is useful. And then, of course, through the discovery of novel adjuvants. And of course, we're all familiar with adjuvants as being very useful in potentiating the activity of vaccines, but also in terms of antibiotics. So, of course, clavulanate is a very commonly used antibiotic adjuvant in clinically in Augmentin, and many of us will have taken Augmentin for various infections. So clavulanate will of course tie up beta-lactamase enzymes that tend to make a lot of pathogens resistant to beta-lactam antibiotics. And so when you use clavulanate with a beta-lactam antibiotic, you can get enhanced activity. So finding new adjuvants or new combinations of antibiotics, perhaps with adjuvants, could be very useful as a way of helping to address the AMR crisis, particularly if we have some understanding about how these new adjuvants are working. And so we're kind of interested in that idea that, you know, not just identifying maybe an adjuvant, but also hopefully understanding how it might work. So our research then is focused on Staphylococcus aureus and MRSA. Of course, Staph aureus is a very important pathogen, both in healthcare settings and in the community. You know, we still have up to 20% mortality in patients with MRSA infections, despite the availability of antimicrobial chemotherapy, which should work. So it's still a very difficult infection to treat. We know that there's resistance to every licensed anti-staphylococcal drug. So that's, of course, a problem. But all the while, you know, beta-lactams remain the first-line therapy for Staph aureus infections, even if most beta-lactams, now there are specific beta-lactams for MRSA, but most beta-lactams are ineffective against MRSA. So this is kind of an important area. So MRSA then, you know, is of course a methicillin-resistant Staphylococcus aureus. I don't want to talk too much about the mechanism of this. Some of you may be familiar with this, but for those of you who aren't, very briefly. So if we look at Staphylococcus aureus, there are 4 penicillin binding proteins that are very important in cross-linking the cell wall during cell wall biosynthesis. When the bacteria are exposed to beta-lactam type antibiotics, including methicillin, the antibiotic binds to these 4 penicillin binding proteins and inhibits transpeptidase activity. Now, PBP2, of course, is a bifunctional enzyme. It has transglycosylase and transpeptidase activity. So the transglycosylase activity remains functional, but the transpeptidase activity is inactivated. And of course, that has the effect of weakening the cell wall, ultimately leading to lysis. In an MRSA, the bacteria have picked up a new penicillin binding protein called PBP2A, or sometimes PBP2 prime. This transpeptidase enzyme is not inhibited by beta-lactam antibiotics, including methicillin. And so in combination with the transglycosylase activity of PBP2, it can continue to synthesize and cross-link a cell wall, allowing the cells to grow. So in essence, that's how MRSA is resistant to beta-lactam antibiotics. Okay, so from our point of view then, just going back, and again, if you'll indulge me, so going back maybe over 20 years when I worked, and Jose mentioned I worked in Royal College of Surgeons, and the teaching hospital for RCSI was Beaumont Hospital in Dublin. And at that time, we collected lots of Staph aureus clinical isolates, you know, so they were methicillin sensitive Staph aureus and methicillin resistant Staph aureus. And at that time, my primary interest was in biofilm. And of course, biofilm associated infections are very antibiotic tolerant, so there is a connection to antibiotic resistance. But we took these clinical isolates and we looked at their biofilm and virulence mechanisms, and we did a lot of work on this over many years. But in terms of biofilm, one of the things that we noticed many years ago really was that MRSA and MSSA make different types of biofilm. So MSSA will make a polysaccharide type biofilm. And so you can see this here on a scanning electron micrograph. It produces an extracellular polysaccharide called PIA, or polyacetyl glucosamine. So the bacteria are embedded in this plume of extracellular polysaccharide, and they stick to surfaces. But interestingly, when we were looking at clinical isolates of MRSA, what we discovered was that even though they have the genes to make this polysaccharide type biofilm, they don't use them. Instead, they use surface proteins, in particular the fibronectin binding proteins, and also extracellular DNA to make a different type of biofilm. So we saw that antibiotic resistance was affecting the type of biofilm that these clinical isolates of Staph aureus were making. So this was kind of an interesting observation, and it kind of shifted the focus of my research, excuse me, somewhat away from biofilm phenotype to being interested in antibiotic resistance. So thinking about that, so of course then we became, because we saw that there was this difference in the biofilm phenotype between a methicillin susceptible Staph aureus and a methicillin resistant Staph aureus, we started to think about the MRSA phenotype. And of course, methicillin resistance in Staph aureus is somewhat complex. And so this is a kind of a slide that I like to use to kind of explain the phenotype. So if we start off with a regular Staph aureus or a methicillin susceptible Staph aureus, it lacks the MEK-A gene. The MEK-A gene is the gene that encodes that penicillin binding protein 2A, that transpeptidase enzyme that can continue to function in the presence of beta-lactam antibiotics. Interestingly, when the bacteria acquire the MEK-A gene, either naturally or perhaps on a plasmid, which is the experiments that we did, what happens is that only some of the cells within the population express high levels of resistance to methicillin or other beta-lactams. And so essentially what you get is what's called a heterogeneous phenotype, where there's a mixture of highly resistant cells and cells that are generally quite susceptible. Now, those cells, those populations, those heterogeneously resistant strains can become homogeneously resistant if you expose the bacteria to oxacillin. Okay, so that then selects for what are called accessory mutations or potentiator mutations that help to make the bacteria more resistant to beta-lactam antibiotics. And so here in these homogeneously resistant strains, all of the cells in the population are resistant to high concentrations of the antibiotic. So they have the MEK-A gene, but they'll also have other mutations on the chromosome that are enabling that high-level resistance phenotype. So we can see here that typically, actually, the homogeneously resistant MRSA strains will be present in hospitals. And generally speaking, without oversimplifying it, in community MRSA strains, they tend to be heterogeneously resistant. So they have the MEK-A gene, but their MICs, although they're resistant, their MICs are lower than the MICs that you would see in the homogeneously resistant strains that are found in hospitals. And also just to mention, of course, that that exposure of a heterogeneously resistant Staph aureus MRSA toxicillin, it will select for these mutations that enable high levels of resistance, but also the bacteria will respond to the oxacillin, and you'll get the MECR1 protease will detect the beta-lactam antibiotic. It'll cleave the MEK-I repressor. That repressor dimerizes and sits on the MEK-A promoter, turning off the MEK-A gene. So in the presence of oxacillin, that repression is relieved. You get an increase in MEK-A expression, and that also increases resistance. So that's a classic way that, of course, any bacterium will increase the expression of a gene that it needs to withstand an extracellular stress. So I'll just mention that at this point as well. So it's a somewhat complicated phenotype. It's not just a matter of a Staph aureus picking up the MEK-A gene and all of the cells becoming resistant. It's not like that. When it happens, you get some of the cells will be highly resistant, but you get this heterogeneous population, and you need additional mutations to give rise to high levels of resistance. So in order to kind of study initially the biofilm phenotype and the effect that antibiotic resistance or methicillin resistance was having on the biofilm phenotype, we first of all needed to construct isogenic strains in the laboratory. And the reason for that was, of course, that if we took a clinical MSSA isolate and a clinical MRSA isolate, they were completely different from each other. They're genetically not the same. So we couldn't really make any meaningful comparisons between the biofilm phenotypes of two clinical isolates. So what we set about doing was taking a laboratory strain of Staph aureus 83254. We introduced the MEK-A gene on a plasmid, on a very low copy plasmid. It was actually technically difficult to do that. It didn't like to pick up that plasmid. But when we introduced the MEK-A gene, then we generated a heterogeneously resistant strain of MRSA, which had a low MIC. And then in order to convert that strain into a homogeneously resistant MRSA strain, we exposed it to oxacillin, generated the homogeneously resistant strain, and now we had isogenic methicillin susceptible and methicillin resistant strains. And I'll just mention here that we sequenced this homogeneously resistant strain that we generated in the laboratory and identified a GDPP mutation which controls cyclic AMP signaling. And I'll kind of come back to that later in the seminar. So once we had these two isogenic strains, we were then able to, the idea was that we wanted to compare their biofilm phenotypes. Okay, so we could see that in the laboratory, the MSSA strain made a polysaccharide type biofilm, and excitingly for us, when we converted that MSSA strain into a homogeneously resistant MRSA strain, we switched that biofilm phenotype to a protein eDNA type biofilm, which was exciting. So we were able to kind of recreate what we were seeing in the clinical isolates in isogenic laboratory strains. And then we wanted to ask the question, well, does this matter? So if the biofilm phenotype of a Staph aureus changes when you make it resistant to methicillin, you get a different biofilm phenotype, does it matter? So to ask that question, we used a mouse model of device infection, where a little piece of catheter is implanted under the skin of a mouse and infected with the bacteria. So we would have infected the mice with either the MSSA strain, the parental strain, or the MRSA derivative of that laboratory strain. And the idea was to compare how much biofilm formed on that piece of catheter to kind of get a sense of whether it mattered what type of biofilm the Staph aureus strains were producing. And surprisingly, what we found was that the mice infected with the MRSA strain survived much better than the mice infected with the parental strain, which was methicillin susceptible, which was a very unexpected finding. And in fact, most of the mice that had been infected with the parental strain at 2 different inocula died within 2 to 3 days, whereas most of the mice infected with the isogenic strain that was now methicillin resistant actually survived. So that was a very unexpected result. And it kind of showed us that methicillin resistance, the expression of methicillin resistance in Staph aureus, was affecting the biofilm phenotype and also affecting how virulent the bacteria were, okay? And I won't talk about it too much, but this attenuation of virulence in MRSA that are highly resistant to the antibiotic is actually to do with a significant downregulation of the AGR locus. And the AGR locus is really controlling the production of extracellular enzymes and toxins in Staph aureus. So when the bacteria become highly resistant to methicillin, they actually downregulate the AGR locus. Okay, so there appears to be this kind of a balance between how resistant the bacteria are and how virulent they are. So when they become highly resistant, they actually downregulate their virulence. And that's kind of interesting, because from my discussions with some clinicians, they'll often talk about the fact that in hospital environments, patients with MRSA infections, those infections are very difficult to manage, but actually they're not necessarily that virulent. So the patients aren't necessarily sicker. In fact, sometimes they're not as sick as they might be with a Staph aureus, which is not methicillin resistant, but that's a bit anecdotal. So arising from that, what we then thought about, well, we see now that methicillin resistance is controlling virulence, okay? And so we kind of asked the question, does that mean that we can actually control virulence by controlling resistance? And the way that we can control methicillin resistance is to actually expose the bacteria to a beta-lactam. In this case, we used oxacillin. And again, you might recall just a moment ago, I talked about when you expose MRSA to a beta-lactam like oxacillin, they can detect the antibiotic and upregulate the expression of MEK-A, which makes them more resistant. And of course, when they become more resistant, then they downregulate their virulence. And so we did quite a lot of work on this. And in the end, we did see that when we infected mice with a community MRSA strain, and a community strain tends to have an intermediate level of resistance, that resistance can be kind of increased or decreased. Okay, and I'll talk more about that. But in that community MRSA strain, this was J2, when we infected the mice with J2, did a bacteremia model, and then treated those mice with 2 different concentrations of oxacillin. And again, the bacteria, of course, are resistant to oxacillin and other beta-lactams. What we found was that we could significantly attenuate virulence. So you can see here on the left, this is just a kidney taken from a mouse infected with the J2 strain of MRSA, and you can see these very visible abscesses on the kidney of the mouse. Whereas here, when we've used, this is a low concentration of oxacillin, and here is a higher concentration of oxacillin, both within the therapeutic range that you could achieve in patients using something like flucloxacillin. And you can see that far fewer of the mice had abscesses. Only 4 of the 10 mice had abscesses when they were treated with a low dose of oxacillin, and at a high dose of oxacillin, none of the mice had abscesses. And you can see that the kidneys looked much healthier. They're kind of perfused and much deeper color than the kidneys from mice that were untreated. So that kind of showed then that we could kind of, by controlling the levels of resistance to methicillin, we could actually control virulence. So it's kind of counterintuitive, of course, but you know, it shows that if you have an MRSA infection, actually using oxacillin, even though it won't overcome the infection, might have the effect of downregulating virulence, and perhaps using that antibiotic in combination with another drug that can hopefully, something like daptomycin, perhaps that might eradicate the bacteria. You might make them less virulent and hopefully then also help eradicate the infections. So that data, you know, was kind of interesting, and it kind of highlighted, excuse me, the importance of genes and pathways that are controlling the resistance phenotype. So, for example, we had, excuse me, we had shown that when we were trying to construct an isogenic, homogeneously resistant strain, that when we expose the bacteria to oxacillin, we'll get mutations elsewhere in the chromosome. And one of those mutations was in GDPP. That's controlling how resistant the bacteria are. And of course, when you're controlling how resistant the MRSA are, you're also having an impact on biofilm and on virulence. So we started to become interested then in the antibiotic resistance phenotype and how the methicillin resistance phenotype in Staph aureus is very much a regulated phenotype, which can have lots of knock-on effects. So, okay, so moving on from that, so at that point then we'd actually at this point become really more interested in that resistance phenotype, and we were kind of moving away a little bit from biofilm and starting to think about antibiotic resistance, which is really what I'm going to talk about in this talk. And so we started to become interested, again, just to recap again, so in Staph aureus, when the bacteria acquire the MEK gene, typically it's on an element called SCC-MEK, you get this heterogeneous phenotype, and then you get other mutations on the chromosome giving rise to higher levels of resistance. So we wanted to then think about, could we identify what these other mutations were? So we knew about GDPP, but we believe, and other research groups had shown that other accessory mutations could also contribute to resistance. So we wanted to know what those other genes and pathways were that were controlling the resistance phenotype. And the reason we wanted to do that was because if we could understand how the resistance phenotype is being controlled, maybe we can find new ways to control resistance. Okay, and so to do that, then we took advantage of the Nebraska transposon mutant library, which had been constructed by Paul Fay and colleagues in Omaha. And in this library, every non-essential gene in the MRSA chromosome has been mutated. And of course, this then allowed us to screen all of those individual mutants for mutations that were affecting how resistant that strain was. Okay, so we did these disk diffusion assays, a very straightforward assay where we would, on this plate here, you can see in the photograph, we have 4 different strains. We have the wild type and a number of other strains. And you can see here that you get a certain zone diameter for the wild type strain. It's resistant, in this case, to cefoxitin, but you can also get mutations, not in MEK, but in other genes, which might make the bacteria more resistant, more susceptible, I should say, or more resistant, okay? And by identifying what those mutations were, we were able to then study other genes and pathways that are indirectly controlling the resistance phenotype. And again, the reason we wanted to do that was to try and find new ways to control how resistant the bacteria were. So we found lots of interesting mutations and pathways that control resistance. But in this talk, I'm going to talk about mutations affecting purine metabolism. And specifically, what we found was that mutations in the de novo purine biosynthesis pathway, or in particular the purine salvage pathway, increased resistance to beta-lactams and MRSA. So that is, when you have a mutation that affects purine metabolism, the MRSA become more resistant. Now, you might ask, why are we interested in mutations that make MRSA more resistant? But I'll come to that. Okay, so specifically, among the first mutations that we identified were in these genes here encoding 3 purine permeases, so NUPG, PBUG, and PBUX. So they transport guanosine, inosine, and xanthine, and we found that they can make the bacteria, particularly the NUPG and PBUX mutations, will make MRSA more resistant to beta-lactams. So we did a lot of work to confirm this. You know, we would transduce those phenotypes from the library mutants back into fresh J2, the wild-type strain, or indeed into other MRSA strains. We would also complement the mutations, with the wild-type genes just to make sure that the phenotypes were reliable. And initially, in our initial screen, we identified this NUPG gene, which is a guanosine transporter. We also identified a mutation in DOD2, which is a nucleoside phosphorylase, so it's a little bit downstream in purine salvage pathway, and it also increased oxacillin resistance. And subsequently, we had some HPT mutations from the purine salvage pathway that also increased oxacillin resistance. So all of these mutations, again, and you can see that here on this graph, just increased the MIC of the mutants to oxacillin, and the phenotypes were reliable and verified. So again, and just to go over that again, so again, if we took the purine operon, which is the operon responsible for de novo synthesis of purines, all mutations in that operon, except pure H, which is slightly anomaly, increased the oxacillin MIC from 64 to 128 microgram per mil in that MRSA strain. And convincingly, for me at least, when you mutate the pure OR repressor, which has the opposite effect of a pure operon mutation, so it increases pure operon expression, you get the opposite effect on oxacillin susceptibility. So you reduce the MIC from 64 down to 32. We also knew that the pure operon was strongly downregulated under oxacillin stress, and that was work that we had done some years ago, which is kind of interesting. So the bacteria appear to be kind of responding to oxacillin stress. And one of the things that they do is actually downregulate the pure operon, and that seems to be contributing to increased resistance. And then, of course, we know then that increased levels of these purine signaling nucleotides, I've talked about cyclic AMP, but also the stringent alarmone PPGPP, when you get increases in the levels of those signaling nucleotides, that correlates with increased resistance. In fact, that's required for increased resistance to beta-lactam antibiotics. Okay, and other groups have shown that if you block the ability of the bacteria to mount a stringent response, in other words, to produce lots of PPGPP or indeed cyclic AMP, that will have a negative effect on how resistant MRSA is. So all of these data were already strongly implicating purine metabolism in the control of MRSA, oxacillin, or beta-lactam resistance. So, and again, of course, one of the things that when we look for mutations that affect resistance, often one of the first things we look at is to see whether it's having an effect on the expression of MEK or on the levels of PBP2A, which of course is the transpeptidase enzyme that mediates resistance. So we looked at our NUPG and DOD mutants initially. So they're the two mutants that are showing a higher level of resistance to beta-lactam antibiotics. And we showed that there was no change in the levels of PBP2A in these mutants, okay? Which again indicated that this change in susceptibility to beta-lactams was independent of changes in MEK expression or PBP2A expression. So that's just something we wanted to rule out. So that, of course, made it more interesting to us because it suggested that there was some sort of different biological response at play, and it wasn't just about controlling MEK expression. So, okay, so of course, all of that is kind of interesting, but those mutations were the NUPG and the DOD2 and other purine operon mutations were all making the MRSA more resistant, okay? And of course, the last thing we need is to make MRSA more resistant. What we need to do is make MRSA more susceptible. So Chris Campbell in my lab, was a PhD student at the time, had this idea that, well, if blocking purine transport, if you block purines being salvaged by the MRSA, if that makes them more resistant, what happens if you do the opposite, if you expose them to high concentrations of purines? It was a great idea, and it was a great experiment. And you can see here on the left, so here's our J2 strain growing on Muruhinton agar with 4 different beta-lactam antibiotics. And you can see that this strain is particularly resistant, so you get very small zones of inhibition around the antibiotic discs. But when you take the same agar and you supplement it with guanosine, so one of the purine nucleosides that we've been using, you can see that you get this tremendous increase in beta-lactam susceptibility. It's a very exciting result. I still love to show this plate. It's really nice, and we show it all the time. So that means that you can essentially, and again, it shows how, screening a transposon mutant library, looking for mutations that affect resistance, can reveal interesting things. And sometimes you can take the original observation and sort of flip it around a little bit and find something potentially interesting. So again, so we kind of then lots of work to kind of verify this. We know that, exposing or growing MRSA in the presence of guanosine or indeed xanthosine, another purine nucleoside, will increase their susceptibility. So here, for example, this is the wild type J2 strain. Here it is just growing in the presence of guanosine. When you add a little bit of oxacillin, it just can't grow. Similarly, if you take xanthosine, a related purine nucleoside, same thing happens. Even at very low concentrations of oxacillin, it's no longer able to grow. But remarkably, okay, and I'll come back to this, when you take, so we started to look at different purine nucleosides. Guanosine, xanthosine made the bacteria more susceptible, but when we took another purine nucleoside, adenosine, we got the opposite effect. Adenosine actually increased resistance. And again, I love that result. So it's kind of interesting because it's kind of showing that depending on how you manipulate purine metabolism, you can make the bacteria more resistant or more susceptible. So adenosine makes the bacteria more resistant. We did these experiments, again, I don't have to go through these in too much detail. We did these experiments also not just in Muller-Hinton media, but also in chemically defined media. And again, you can see here up on the top left, when we grow the bacteria in chemically defined media with guanosine, they grow happily. If you add even a tiny concentration of oxacillin, they can't grow. In contrast, if you add adenosine, you start to see that they become more resistant and they now can grow even at low concentrations of oxacillin. We did little experiments where we added we took away adenosine and guanosine. We see some resistance. If we have adenosine plus guanosine, you can start to interfere with the activity of guanosine in potentiating oxacillin, and you start to get some resistance. But I won't go into too much detail on this. But we did lots of experiments to verify that these phenotypes were real. We also did checkerboard titration assays. Many of you will have done these. Here we just have an increasing concentration of oxacillin along the top, and down here an increasing concentration of oxacillin just along on the left hand. And you can kind of see that as the concentration of guanosine increases, the MIC of the wild type bacteria goes from 64 down to about 1. And of course, at an MIC of 1, this would now be a methicillin susceptible strain. It wouldn't be an MRSA at all. So that was kind of exciting. And this is just to kind of, again, because I'm talking about different purine nucleosides and how they're affecting resistance. So in this figure on the left, it looks a little bit complicated, but I'll just go through it. This is just a summary of the purine salvage pathway in Staph aureus. And just here, over here on the left, this is just the membrane. Here I've shown a few different transporters that are involved in taking up purines. So NUPG will take up guanosine, PBUX will take up xanthosine, PBUG here, or NUPG, and then PBUG will take up inosine. And so what we found was then that when we expose the bacteria, and I've kind of talked about this, I've shown it on growth curves, but if you measure the MIC, when you grow the bacteria in the presence of guanosine or xanthosine, you reduce the MIC from 64 down to 1 or even down to 0.5, so they're completely susceptible. So xanthosine and guanosine are coming in here into what's called the GTP branch of purine metabolism. So ultimately, they can be used to make GTP, okay? If we use, if I just move on, and if we used, sorry, just move on. So when we used adenosine, then we got the opposite effect. And I kind of already alluded to this. So adenosine, it feeds into the ATP branch. We're not actually sure what the transporter for adenosine is yet. It'll come into the ATP branch and be used to make ATP. And you can see here that the MIC goes from 64 up to 256. And then if we use, then finally, if we use inosine, we get very little change in the MIC. So inosine comes in here, comes into inosine monophosphate, which can be fluxed to either the ATP branch or down to the GTP branch. And so you don't actually see much difference. And of course, it's another result that I love. I always talk about this result because adenosine will make the bacteria more resistant. Guanosine or xanthosine will make them more susceptible. Inosine, which can be fluxed either to the ATP branch or to the GTP branch, really doesn't have any significant effect at all. And just when I'm showing this figure, just to also mention, because I've talked about the signaling nucleotides that are controlling resistance. So of course, cyclic AMP is up here and PPGPP is down here, okay? these two interact with each other. I don't want to go into too much detail about that, but of course they are derived from purine metabolism, and so they're important. We already knew that they were important for controlling the resistance phenotype. So again, we're just manipulating this purine salvage pathway in ways that can either make the bacteria more resistant or more susceptible. So, and again, because we have the Nebraska transposon mutant library, we have various mutations in these pathways. So and again, we pursued these experiments to give us more and more confidence that what we were doing was real. So for example, when we took a DOD2 mutation, okay, so DOD2 encodes a nucleoside phosphorylase, which converts guanosine to guanine. So in this case, when we use guanosine to try to make the bacteria more susceptible to oxacillin, it didn't work. Okay, and the reason it didn't work was because even though the guanosine can be taken up by the bacteria, that enzyme that converts it to guanine and downstream to the GTP branch of purine metabolism doesn't work, so we don't see any effect. However, in a DOD2 mutant, xanthocene did make the bacteria more susceptible, and that's because xanthocene comes in via a different transporter, can be converted to xanthine, and can bypass the DOD2 mutation here so that you can potentiate the activity of oxacillin. Similarly, we also had 2 HBT mutations, which we obtained from David Heinrichs in Canada. And we also saw in this case that guanosine couldn't potentiate oxacillin in these HBT mutants, but again, xanthocene could, because again, it'll bypass this mutated branch of purine salvage pathway. And then for a DOD1 mutant, both guanosine and xanthocene could resensitize that mutant. But again, this was just giving us more and more confidence that, you know, these mutations in the purine salvage pathway were controlling resistance, and that we could manipulate the resistance phenotype by interfering with this pathway. So, of course, then one of the major determinants of whether the guanosine can get into the cell to resensitize the MRSA is whether it can go through NUPG. So here's NUPG here, which takes up guanosine. So, of course, in a NUPG mutant, guanosine doesn't work anymore. So in a NUPG mutant, even when the bacteria are growing at low concentrations of oxacillin or even high concentrations, the mutant grows well, okay? Whereas xanthocene can resensitize a NUPG mutant because, again, xanthocene isn't coming through NUPG, it's coming through PBUX. So when you grow a xanthocene mutant, especially at higher concentrations of oxacillin, you can again resensitize the bacteria to the beta-lactam antibiotics. So again, all of this is kind of giving us more and more confidence that these phenotypes are real. And of course, one of the first things that you might ask when you hear about something like guanosine or xanthocene, you know, making the bacteria more susceptible to an antibiotic, one of the first questions we asked, well, was it having a negative effect on growth of the bacteria? Okay, and we didn't see that at all. Okay, so in fact, here, if you just look at growth of J2, this is the wild-type MRSA strain. So this is just growing in MHB, and you can see here in MHB these kind of light blue. This light blue graph here, you can see that it grows pretty well in MHB, obviously. But when we add guanosine, not only does it not negatively affect growth, in fact, it grows better. So you can see this very significant increase. So the guanosine appears to promote growth, which was a little bit surprising, whereas the adenosine appears to actually negatively affect growth. And this is based on cell density spectrophotometrically. And then we found that xanthocene didn't really do much. It was kind of the same as the wild type. But what this kind of showed was that there was no effect, certainly no negative effect of guanosine or xanthocene on growth. And if anything, it was promoting growth, possibly because there's ribose sugars associated with these purine nucleosides, which can actually be metabolized by the bacteria. We were a bit surprised by this very significant increase in growth by guanosine. And so we also looked at colony-forming units, so enumerated colony-forming units. And actually, when we did this, we found that there wasn't really that big a difference. So most of the graphs overlapped with each other. We didn't see that increase in growth promoted by guanosine. So what that kind of suggests, of course, is that here that the cell size has changed. So one of the reasons you might get increased cell density at OD600, for example, is because the cell size has changed. And in actual fact, I'll come back to that because that's what turned out to be the case. And again, just on this one, I have it. You can see here that in a NUPG mutant, we really didn't see much happening because, of course, you know, NUPG mutant cannot take up guanosine, so you don't see this increase in cell density, at least measured at OD600 in an NUPG mutant. And again, the enumeration of colony-forming units was the same. But ultimately, what this suggested was that guanosine certainly didn't increase MRSA susceptibility to beta-lactams by having a negative effect on growth. And if anything, it may even have a positive effect on growth. We then also showed, we looked at several, most of our experiments up to this point had been looking at this MRSA strain here, J2. We looked at several other diverse MRSA strains that we have in our collection. And for the most part, we found that growing the bacteria in combinations of oxacillin plus guanosine plus its adjuvant guanosine would have a significant effect on reducing the MIC. So sometimes it might only go from 64 to 32. Often it would go from something like 256 down to 16. So it varied a little bit, which I'll come back to, because even though we're very happy with the therapeutic potential of this, the bacteria have the potential to become resistant to this combination of the antibiotic and the adjuvant. And perhaps in some of these clinical isolates, there are some mutations already in place that are giving the bacteria resistance. So for example, with col, which is a well-known strain, you can see that we weren't really able to increase the susceptibility of col to oxacillin using guanosine or indeed xanthocene, but I'll kind of come back to that. But one of the interesting things, we did include a Staph epidermidis strain, in this case RP62A. We were able to significantly make it more susceptible. And we also used our laboratory Staph aureus strain, MSSA strain A3254. And actually, we were able to reduce the oxacillin MIC for this strain as well. So, of course, that's MEK-A independent. There's no MEK-A in that strain, which is kind of interesting. So that sort of suggests that, you know, that the purine nucleosides working as adjuvants might have MEK-A dependent and MEK-A independent roles, which could potentially broaden their usefulness. We also, and again, these are kind of busy slides. You don't need to show you this data, but essentially we showed that guanosine and xanthocene can synergize not just with oxacillin, but also other beta-lactams, so methicillin, nafcillin, cefoxitin, you know, penicillin G, etloxacillin. We used a bunch of other beta-lactams, not exhaustive, but, you know, several other beta-lactams, and found that we could see that synergy between guanosine or xanthocene and those beta-lactams. But when we looked at other just random antibiotics, and in this case, we initially used tetracycline, vancomycin, because it also targets the cell wall, chloramphenicol or gentamicin, we didn't see any effect. So at this point, our data was kind of suggesting that it was specific to the beta-lactams, okay? And it wasn't, certainly not affecting, you know, antibiotics that were targeting, for example, protein synthesis, or indeed vancomycin, which is also targeting the cell wall. So that was all good. But as I mentioned, it's not all good. Some of the clinical isolates were resistant to that therapy, so they didn't show that increased susceptibility to combinations of oxacillin and guanosine. And so to kind of investigate, and again, to investigate that, and also to investigate how the bacteria might become resistant to these combinations, we raised suppressors. So we looked for mutants that were resistant to oxacillin and guanosine used in combination. And we identified interesting mutations, many of which kind of supported some of the data we already have. So for example, we got suppressor mutations arising in NUPG, in HPT, and also in PRS, which is involved in de novo purine biosynthesis. And those mutations made the bacteria more resistant to the oxacillin-guanosine combinations. So that's not good. And that, of course, suggests that even if this was to be used therapeutically in a clinical context, you could get mutations giving rise to resistance. But one of the things that, you know, when we were investigating this, we also we don't just measure susceptibility using, for example, checkerboard assays or disk diffusion assays. We also use kill curve assays. And in this case, you can see here that when we have our wild type J2 strain, and we have a suspension of cells in either MHB or an MHB with oxacillin, with the combination of oxacillin and guanosine. And in this case, we increased those concentrations of the oxacillin and the guanosine, still therapeutic range for oxacillin, and as low a concentration of guanosine as we can get it. And you can see here that in the red one, that when we use higher concentrations of oxacillin, we tend not to see these suppressors arising. So we tend to be able to, if we take a suspension of cells, expose them to oxacillin and guanosine at a high enough concentration, we can kill them and they don't recover. When the concentrations of oxacillin are a little bit lower, so for example, in this green graph here, you can see that after about 12 hours, you get these suppressor mutations that make them resistant, okay? And again, this isn't unexpected because again, as I talked about at the very start, we know that these mutations elsewhere on the chromosome arise, well known to arise, that increase resistance to methicillin and MRSA. And so you can see that happening here. But when we increase the concentration of oxacillin, we think even within a therapeutic range, we might be able to stop that from happening, which would make this more therapeutically useful. So, okay, so what else is happening when you grow the bacteria, the MRSA, in the presence of oxacillin and guanosine? We know that the cell size increases a lot. So these are just some nice microscopy images. And you can see here's the wild type MRSA. Here's the MRSA grown in guanosine in the presence of oxacillin, and then oxacillin and guanosine. And you can see here that visibly, you can see that the cell size is dramatically increased when the MRSA is grown in the presence of oxacillin and guanosine. So it had already been reported that oxacillin does increase MRSA cell size, but when you use the combination of oxacillin and guanosine, you get that dramatic increase in cell size. That's very significant because we had already shown that mutations in GDPP, which control cyclic AMP signaling, are important for oxacillin resistance in MRSA. And we know that the cyclic AMP signaling is controlling osmotic stress in the bacteria. And we see that now that the bacteria are becoming bigger in the presence of oxacillin and guanosine, which is again pointing towards cyclic AMP signaling as being altered in the bacteria. And you can see that, I'm not sure if I have time, but again, you can see that here's a cell, an MRSA cell grown in the presence of oxacillin and guanosine compared to the wild type cells up here on the top left. And you can see that, you know, they're really gigantic compared to the wild type cells. So you're getting that altered osmoregulation in these bacteria, which is probably consistent with the cells undergoing osmotic lysis in due course. So, okay, so what ultimately, and I don't know if I have time to go into this in too much detail, but ultimately what we did was then we collaborated with Angelika Grundling's group in Imperial College, and we were able to show that when we expose MRSA to exogenous guanosine, we're able to regulate cyclic AMP in a NUPG dependent manner. And again, I have a lot of data here. I don't think I'm going to go through it. Just to show here in this middle graph, when we expose the bacteria to oxacillin and guanosine, we get this dramatic, significant reductions in the levels of cyclic AMP. And it's kind of related to NUPG. So NUPG has to take up the cyclic AMP, the guanosine, in order to see that happening. But as I mentioned earlier, that cyclic AMP levels need to be high in order for the bacteria to be resistant. And you can see here that when we expose the MRSA to oxacillin and guanosine, the cyclic AMP levels are pushed down. And that's consistent then with the bacteria being more susceptible to the beta-lactam antibiotics. And indeed, it's completely consistent with that very dramatic increase in cell size. So just to mention that. So we believe the data is pretty tight that cyclic AMP is at least playing part of the explanation for why the bacteria are becoming more susceptible to oxacillin when you expose them to purine nucleosides. And again, so guanosine is interfering with that normal activation of cyclic AMP that's needed for high-level beta-lactam resistance. And again, so Merva Zedin, who worked on this project during her PhD in Imperial College with Angelika, Merva had shown that other mutations that directly control cyclic AMP, so for example, mutations in DAC-A, which is the enzyme that makes cyclic AMP, or in GDPP, which is the enzyme that turns over cyclic AMP. When you mutate DAC-A, you get a decrease in oxacillin resistance. When you mutate GDPP, you get a very significant increase in oxacillin resistance. So that correlation between cyclic AMP and methicillin resistance is very well established. And then just here to show that, of course, I've been mentioning that adenosine has the opposite effect. So adenosine doesn't increase the susceptibility. In fact, it makes them more resistant. So when we looked at the effect of adenosine on the bacteria, we didn't see any change in cell size, no significant change in cell size when the bacteria were exposed to adenosine, or even a combination of oxacillin and adenosine. And also we didn't see adenosine having any effect on cyclic AMP levels. So we can sort of again, separate the effects of adenosine from the effects of guanosine or indeed xanthosine. Okay, so I'm kind of running a bit short, but I want to just move on a little bit because so our data essentially up to now is showing that really potent activity of these purine adjuvants in increasing susceptibility to beta-lactams. But more recently, we've discovered that it's not just beta-lactams that we can potentiate with these purines, okay? And Aaron Nolan, who's done a lot of the work on this, had done some very nice metabolomics and proteomics. So these are some nice volcano plots looking at the proteome of MRSA in bacteria grown in the presence of oxacillin versus bacteria grown in the presence of oxacillin and guanosine. And you can see all of these proteins which are differentially regulated. He also did some metabolomic analysis, and this is just some of the metabolomic analysis for purines and pyrimidines. And what I want to kind of point out from this is we already knew, of course, that guanosine will come in and affect purine metabolism, but what this data revealed that there were also effects on pyrimidine metabolism. And so we can see up here that this purine intermediate AI-CAR, which is a very important intermediate in purine biosynthesis, is significantly downregulated. But in the metabolomic analysis and indeed in the proteomic analysis, when you look at the pyrimidines, we saw this dramatic effect on thymidine. Okay, it really jumped out. Thymidine was dramatically downregulated by guanosine or by oxacillin and guanosine. So that was, of course, really interesting. So, and just thymidine, it wasn't all of the pyrimidines, primarily thymidine. We didn't see anything happening, just to mention, with adenosine. Again, you can see that the proteome of the bacteria exposed to adenosine and oxacillin versus oxacillin alone is dramatically different than what we would see with guanosine. So again, it's just separating out the effect of those purine nucleosides on the physiology of the MRSA. So thymidine, so here's thymidine. So this is, again, a complex graph. I don't really want you to go through everything, but we became interested in thymidine because, of course, and we know that thymidine is taken up by this transporter here called NUP-C. It's very important for DNA synthesis. Pyrimidine metabolism also provides intermediates for cell wall biosynthesis. But thymidine levels were significantly downregulated by guanosine. So we started to look at that, and we started to think about, if thymidine levels are reduced by guanosine, can guanosine potentiate the activity of other antibiotics that are targeting thymidine or indeed pyrimidine biosynthesis? And so we started to look at sulfamethoxazole and trimethoprim, the antifolate antibiotics. So they're involved in producing folate for Thia, which converts DUMP to DTMP, which is an important intermediate for DNA synthesis. Okay, and so what we found was that, sure enough, the purine adjuvants did potentiate the activity of these antifolate antibiotics. So that was really cool. So from that metabolomic analysis showing that guanosine would downregulate thymidine, we were able to predict that the guanosine might potentiate these antifolate antibiotics, and that's what happened. Aaron also used an analog of uracil, 5-fluorouracil, which is used actually as an anti-cancer agent, and another related compound, 5-fluorouridine. And again, because that fluxes into purine metabolism and feeds into this Thia reaction converting DUMP to DTMP, we also saw that guanosine would potentiate the activity of 5-fluorouracil and 5-fluorouridine against MRSA. And I'll show you some of that data very quickly. Again, these are just kill curve assays. Again, just hopefully to convince you that this is real. So guanosine, here, for example, these are kill curve assays with our wild type MRSA strain. And just down here, the most dramatic effect. So when we grow the bacteria in the presence of sulfamethoxazole and guanosine, and in this case, we're also adding oxacillin, because we know that the guanosine is potentiating the activity of the oxacillin. It's potentiating the activity of the sulfamethoxazole. Here, we're getting significant killing of the bacteria. When we put them all together, we actually eradicate the bacteria completely. We can't recover any viable bacteria. This is sulfamethoxazole. And again, we see the same sorts of effects with trimethoprim. When we use checkerboard assays, you can see that in the presence of guanosine here, you get a lot more killing by oxacillin and sulfamethoxazole than if you just use them on their own. Similarly, you can increase the activity of oxacillin and trimethoprim against MRSA when you add guanosine. We see this with the pyrimidine analogs, the 5-fluorouracil and 5-fluorouridine. Again, you can see this in checkerboard assays, but here's some nice diffusion assays, which are always very visual. So you can see, for example, here's MW2, an MRSA strain. It's resistant to cloxacillin, 5-fluorouracil, and even a combination of both cloxacillin and 5-fluorouracil. When we add guanosine to the media, now we start to see good inhibition of, or good activity of cloxacillin, 5-FU, and the combination of both. And we can see this to varying degrees with all of the clinical isolates that we look at. You can sort of see, again, here's one of our own clinical isolates from Dublin. Again, you can see that it's very resistant to 5-FU and cloxacillin, 5-FU combinations, and that's dramatically increased when you add guanosine. So again, that's all consistent with the idea that guanosine is down-regulating thymidine levels, and if you come along with other drugs that also target thymidine, you can potentiate their activity. Okay, and again, doing some basic genetics, and again, just very briefly, you can rescue some of these phenotypes by giving the bacteria thymidine. Again, this gives us confidence. So of course, the idea is that, you know, the guanosine down-regulates thymidine. That gives you a phenotype. If you give the bacteria back lots of thymidine in the media, you can overcome these effects. And in fact, that's what you see. So with these wild-type strains and all of these mutants here, when you grow them in 5-fluorouracil, you really have a dramatic negative effect on growth. But if you give them back thymidine, they all grow happily because you're giving them what they need, except in a NUPC1 mutant. And the reason for that is because NUPC1 is required to take up the thymidine from the media. So again, all of this is giving us confidence that this really is, the effect of guanosine on thymidine is very significant. And very briefly, we also have these effects on antibiofilm activity. So Aaron did lots of work on this, showing that you can also increase the activity of oxacillin antifoliate combinations against biofilms if you use guanosine. So, and then finally, just to very briefly mention that one of the consequences of, you know, down-regulating thymidine is that you have an effect on DNA synthesis. You get thymidine starvation, and then that can lead to DNA damage, which activates reactive oxygen species, which leads to membrane damage. Aaron did some very nice experiments with Merva Zedin in the lab, and they showed that, you know, you can see here that, and again, I'll go through this very quickly, but you can see here's the wild-type bacteria growing in MHB. When you expose them to oxacillin or 5-FU or combinations of the antibiotics, you get this dramatic increase in the reactive oxygen species produced in the bacteria. And of course, that's consistent then with having negative effect on the physiology of the bacteria. And you also then get this dissipation of the membrane potential as a consequence of the increased reactive oxygen species. And again, I don't really have time to go into this in too much detail, but here's the wild-type bacteria in MHB. This is for FACS analysis. You can see that when you grow the bacteria in oxacillin plus guanosine, you get this real depolarization of the membrane. Or if you use guanosine, oxacillin, and 5-FU, you really depolarize the membrane. Of course, that's all consistent with improved killing of the bacteria. So we're kind of interested in, again, getting mechanistic insights on the activity of these, you know, these purine adjuvants in terms of overcoming MRSA. Okay, so I'm going to stop there and just try to summarize that with this figure if I can. So essentially what our data are showing, so if you take MRSA and if you leave them untreated, they grow happily, of course. When you expose the MRSA to oxacillin, they survive that treatment. The oxacillin does kind of push down cyclic AMP levels, but it, you know, once the levels of cyclic AMP are at a critical point, they'll become resistant. We know that there's a slight increase in cell size when you grow the bacteria in oxacillin, but again, they can obviously survive that. When we expose the bacteria to oxacillin and guanosine, we get a dramatic increase in cell size. That's associated with a very significant decreased levels of cyclic to AMP. We also know that thymidine levels are significantly decreased. That's associated with membrane depolarization and all sorts of pleiotropic effects, which combined mean that you take resistant bacteria, and once you expose them to guanosine, you get that significant increase in susceptibility to these antibiotics, to the antifoliate antibiotics, to the pyrimidine analogs, and also to the beta-lactam antibiotics. Okay, I'm good. The people who did all this work, sorry, excuse me. Oops, didn't mean that to happen. Okay, so I want to thank the people who did this work. In particular, I want to call out Aaron Nolan and Dr. Aaron Nolan and Dr. Merva Zedin. So Merva now has her own research group in Galway, and she's exploring new frontiers in anti-staphylococcal therapies and indeed other escape pathogens. Aaron completed his PhD recently and has now started a postdoc working on a JPI AMR funded project to continue this work, looking at the ability of the guanosine and other purines to potentiate the antifoliate drugs and the pyrimidine analogs. I also want to mention Chris Campbell, a PhD student several years ago, who did some of the initial work on this. Igor in Angelica Grundling's lab in Imperial, who did a lot of the cyclic AMP measurements. We also looked, Lucy Irwin, we also looked at PPGPP levels. I didn't talk about that data because we didn't see any effect. That work was done by Lucy Irwin in Rebecca Corrigan's work when she was in Sheffield. And then also to mention our collaborators. So Vinay Thomas has always been a great collaborator of ours and helped with a lot of this work and analysis that we're doing. And then for the Purify AMR project, which is again looking at the purine adjuvants to overcome antimicrobial resistance, which is funded by JPI AMR. So Merva is a partner on that with me, as well as Aris Kadulu in Liverpool, who's an expert on in vivo models of infection, and Barbara Cal in Munster, who's interested in different types of clinical infections where we think this could be kind of relevant. And with that, I'll stop, and hopefully I haven't talked for too long, but thank you very much.
Thank you, James. It was just perfect on timing. We still have time for questions. Well, the floor is open for questions. Professor Matty, would you like to start?
First of all, amazing talk, amazing.
We can hear you.
Sorry. I want first of all, Dr. Gara, that was an amazing talk. Being a bacterial geneticist, I really appreciated the clean genetic work you have done. And though I don't follow Steph Warrior's research, it was just a pleasure to listen to all the detailed work. And I hope a lot of the participants here take home that, clean genetics works can give us amazing results. The hard part is constructing the strain. After that, it's an easy one.
That's right.
Anyhow, yeah. So I have a lot of questions. I'm going to go backwards rather than going back to the beginning, because what is fresh in our memory. So the question I had, the first question is that when the cell size increases, so in other words, you're compromised in the division, cell division. So did you find any impact or effect of your treatment or mutations on FTSZ expression?
Yeah, it's a good question. We didn't look. So I don't know, but it could be that for sure we're seeing sort of pleiotropic effects and we're seeing lots of, proteins being up and down regulated. We're seeing lots of metabolites affected. We know that oxacillin alone has pleiotropic effects on the transcriptome. So it would be an interesting thing to look at. But we just haven't looked, so I can't answer it. Yeah.
Okay, thank you. So the thing was, it's the replication rate compromise. I'm trying to understand why it's enlarging, and clearly when you mess with the purine and pyrimidine metabolism, you're going to impact the replication rate, right? And we know that the beta lactams, that they affect the dividing cells, so you're not dividing, right? In this case, so we'll have to go look at the things. So were you able to measure the replication rate?
Well, I suppose not so much replicate. Well, I suppose, we more measured just growth. So we measured cell density and then the enumerated CFUs rather than cell division. Did we look carefully at that? I don't think we looked particularly carefully, but I don't think there was a big difference in the doubling time, if that's what you're getting at.
Yeah, I am.
Yeah, I don't think so. I mean, I think that the thing about cell size is really the cyclic AMP is controlling osmoregulation. And I think that's what's really, once that's distorted, that kind of, at least that's consistent with the, if you like, the swelling of the cells. They're obviously taking on more water. We didn't, when we looked on a microscope, we don't see them bursting, okay, even though they're getting very large, but it seems like, we kind of thought we might, but we didn't really see evidence of that. Whereas in other studies when we saw cells enlarging like that, you would sometimes see debris from the cells lysing. We didn't quite see that.
Yeah.
But presumably those larger cells, are more vulnerable. It kind of makes sense to me, but I know that these things can be complicated.
Yeah. Yeah. I can keep going, Jose. Dr. Puente,
let me
stop
take
me
some of-
whenever you need to. So,
Sure. Let me take some of the audience
Mm.
questions. An anonymous attendee, did you see if similar results occur with other cyclic dinucleotides besides CD, AMP relating to biofilm? For example, CD,
good
GMP.
question.
Yeah, well, there's no cyclic di-GMP in staph, so they don't have it.
Okay.
Like many years ago, in a separate study I did with Mark Gomelski, who's in Wyoming, we looked to see whether Staphylococcus produces cyclic di-GMP. We couldn't find it. Okay, and we looked at some genes that we thought would be involved, and he couldn't find any evidence that they made cyclic di-GMP. And then we did identify at that time, we kind of noted in a paper that we saw this gene that looked like it could have been involved in cyclic di-GMP, but it was kind of, the homology was very low. And we kind of called it GDPP. Little did we know we'd actually stumbled on something really important, but we didn't know what to do with it. And subsequently, it was shown that in Bacillus and then in Staphylococcus and Listeria, that it's really cyclic di-AMP. So the cyclic di-GMP is primarily in the gram negatives. So it's an interesting question, but yeah.
Thank you, James.
Thanks.
Also from this attendee, you mentioned that there might be a MECA dependent and independent roles of guanos in action. Could you please elaborate on what's the MECA dependency of this effect, and if you have tested if any has any altered PBP2A activity?
Yeah, I suppose when I say MECA dependent, I suppose it's just that you see an effect in cells carrying and expressing MECA, and then you see that you can potentiate you can reduce their resistance. And then in cells, even in Staph aureus that doesn't have MECA, you can also reduce their admittedly very low levels of resistance to these beta-lactams. So we don't see an effect. Maybe I should have elaborated on that, but we don't see that. We don't believe there's any effect on PBP2A, certainly expression levels. Activity, could it be? We don't think so. We think even though, so when I say, maybe I may not have put that, it's just that you see it in bacteria that have MECA and that are expressing PBP2A, and also in bacteria that don't. So rather than,
Okay. Does guanosine have effects on daptomycin activity?
No, I don't think we've looked. I'm just thinking, I don't know I'm not sure if Aaron or Merva in the talk with me, but I don't think we've looked at daptomycin. Yeah, so maybe no. But now they could be screaming at me that we have. I don't. If we have, I don't know.
Not that many. What's the mechanism of what's the mechanism of thymidine accumulation and ROS?
Oh, well, so what happens is that when thymidine levels are reduced, you get DNA damage, and that DNA damage is already well known to trigger the accumulation of ROS. So you get this kind of ROS stress, like so. And then once you get ROS stress, you get membrane damage. So, I mean, that would be kind of older literature, really, like not anything that we did. We were following older literature on that, yeah.
Can I follow up with a ROS question, Dr. Puente? Yeah.
Sure, go ahead.
If you grow long enough in ROS, you get resistant mutants. So have you looked at the rise of resistant mutants after extended period in ROS? Extended exposure to ROS?
We haven't, and maybe we should. I know Merva loves screens like that, looking for suppressor mutations or mutants that can tolerate. So we haven't done that. It's another idea. So we could go in hydrogen peroxide or something and just see what happens. Yeah, for sure.
Yes, so I think one way to do is not just it's a continuous exposure.
Okay.
So you do a change of the medium with a fresh ROS.
Yeah.
And then you can select for really good mutants, and then you can go back and find what are the suppressor mutations.
Yeah, no, it's a good question. I mean, I guess when thymidine levels, with a lot of these things, there's going to be lots of effects, of course. So when you start to knock down thymidine, there'll be lots of effects. And, even just in terms of DNA replication is going to be impacted, and gene expression is going to be affected. So lots of things happen. But yeah, it would be interesting to tease it apart. Yeah.
Yeah.
This
Jose,
one
continue. I'll
is,
be right back in a minute.
sure. This is from me, James. Have you tried this treatment in vivo in the mouse model?
We are hoping that's what we're going to do as part of this next grant. So we've tried to do some experiments, and actually one of the challenges is to actually deliver the antibiotic and the adjuvant to the mice, because it's a good question, because the guanosine, we're using 200 microgram per mil. Now, the thing is that the bacteria grow happily. There's no reason to think that guanosine will be in any way toxic. So lots of antivirals and anti-cancer drugs are kind of analogs of these things, but it's actually a solubility issue. So when we try to solubilize some oxacillin and guanosine, they're kind of precipitating. So that's been very challenging. So one of the things that Aaron tried and Dan over in Liverpool was to actually put the guanosine into the drinking water for the mice and then potentially inject them with the oxacillin. But it's a little bit tricky. So this is one of the things that we're going to have to tease out over the coming, probably in the next year or two. But we definitely need to, it's very important to see whether we can translate this into a preclinical model. For sure, it's high on our priority list. Yeah.
And I guess it's also going to be important to see the effect on the microbiome.
Yeah, yes, exactly. Yeah. I mean, I'm always struck by, sometimes in some ways, years ago when I started to think about antibiotic combinations, I always thought, oh, this is great. You could combine two existing antibiotics, and if they work better, great. But actually, as you know, clinically then sometimes you'll get problems. There could be nephrotoxicity or something unexpected when you do that. Or as you say, there could be effects on the microbiome that are unexpected. So while in the laboratory all this looks great and we kind of find it exciting, for sure we need to look at things like that. Unexpected toxicity that maybe we don't anticipate, or microbiome effects. Yeah. So they're on our list. So we have tried some mouse experiments, but so far they've been difficult. So we probably need to think about ways of increasing the solubility of the drugs, which is almost a separate scientific field. But we might need to go down that road. Yeah.
And this is more like a sort of a philosophical question. It looks like this combination of different compounds will be a potential solution for the future to go against AMR. But what your data also reveal is this plasticity that pathogens have to just acquire mutations to become resistant again. So it looks like we are just going to be fighting forever against AMR because bacteria are so way too smart.
Exactly. I always thought that. Can I ask Dr. Gara to unshare so we can, thank you. Yeah. The bacteria is always one step ahead of us.
Sorry, I'm just trying to figure out how to unshare. Sorry, just one.
Okay.
What do I do to unshare?
It's just.
What do I do? Maybe. How do I escape? No, sorry.
Don't worry if you can't. It's not critical.
Unshare. I'm just. How do I? Sorry.
Where you shared, go back to the same spot.
Top, if you go to the top with your prompt, probably should reveal.
Don't worry, James. Let's continue our discussion.
Okay. So normally I can do it. I don't know. For some reason I can't see it now, but
it's at the bottom. It's at the bottom next to the Q&A function.
Okay, I'm not even seeing that now. So.
You put your cursor at the bottom. Yeah.
Okay, cursor at the bottom, and then
you'll see that.
I end the show. Does that help?
Yeah, there you go. Yeah, it's nice to discuss when you see that. So talking about the philosophical questions, maybe this not so philosophical. What is the source of McElliman?
Oh yeah, well, there's been a lot of discussion about that. Well, I think that for people who study this, I think they feel it came from a coagulase negative Staphylococcus species. Somehow
Huh.
it moved into Staph aureus. So I believe that's the most widely held view. So
Okay.
moved from a
So,
coag-neg staph into Staph aureus, and then from there spread. Yeah.
wow. So the other question is, once an individual acquires an MRSA, right, in whatever form, does the microbiome across the body that wherever we find staph get replaced by the new version, or it is only site specific?
Oh, yeah, that's a good question. I'm not yeah, because usually, of course, it'll be in the nares of the nose and some
Right.
other body, in the there's certain areas that will be swabbed. But does it displace? I'm not. That's a good question. Does it displace? I don't know if it does. I guess it's. I wouldn't have thought it displaces the native. It's obviously on the skin and in the nose and the mucous membrane.
Right.
So I don't think it's that it displaces the native flora, but it just kind of establishes itself. Often it's transient. The Staph aureus transiently carried. So I don't feel it displaces. That's my feeling now, without being an expert on that. But doesn't displace, but it's carried for a period of time. And then over time, it can itself be displaced. So does that make sense? I think that's right.
Yeah. I think we're giving you a lot of ideas for undergraduate and graduate projects.
That's right. Yeah, that's right.
So the other question, do you have another one, Jose? You can go.
Yeah, I was just about to question because I probably missed that you mentioned something about it, but I found fascinating that you see down regulation of the AGR genes. It's also MEC-R and MEC-I dependent, the down regulation.
Yeah, so yes, well, ultimately, so what we were able like in very simple terms, if you take a blood agar plate and you put one of the community MRSA strains that has nice hemolysis around it, if you take the bacteria before you spot them onto the blood agar, so if you just grow them in a sub-inhibitory concentration of oxalin, what you'll do is you'll trigger MECA expression, and those, when you spot those bacteria onto the blood agar, there'll be much less hemolysis. So there's a very direct effect. So once you upregulate MECA, you definitely see that down. I believe you see that down regulation of AGR. It's kind of amazing. We kind of looked at that a bit. It's almost like the cell wall has changed when the bacteria, they're making a slightly different type of cell wall. And in fact, Simon Foster's group in Sheffield did some very nice experiments that they published in Science last year, showing the different sort of cell division in an MRSA. And so that different cell wall appears to affect the AGR loop, that sort of the way that they respond to the auto-inducing peptide and turn on the AGR system. So when you have a resistant cell wall, that loop seems to be broken and the AGR system is down-regulated. And so Ruth Massey, we collaborated with Ruth, who's now in Cork, showed that she made some protoplasts where she sort of took away the cell wall, and then she could kind of get the proper activation of the AGR system. So it seems to be to do with somehow when the MRSA become highly resistant and they have a slightly different cell envelope, that AGR system, so it's producing this little quorum sensing peptide which has to be detected, and somehow that doesn't work properly in the MRSA, which is kind of fascinating. It's amazing. So they're very much, the hospital MRSA strains are very much sacrificing virulence for resistance. And the thing is, when I thought about that back years ago when we were doing this, I kind of realized, but in a hospital setting, the MRSA don't need to be virulent. They don't really need to be virulent because, the patients are all vulnerable, they're very immunocompromised, but they do need to be very resistant. So we're using lots of drugs, but they can sacrifice the virulence. And in fact, going back to earlier work that I had done on epidermidis, Staph epidermidis is quite a significant pathogen in immunocompromised patients with lines and catheters and things. And of course, they're really not virulent at all. And so it's almost like the MRSA are saying, well, if they could think, they've decided that they'll sacrifice virulence for resistance. Whereas if you go into the community, in the community MRSA strains, they're highly virulent and they're also resistant, not as resistant, but they've got that balance. And so of course, then the community MRSA strains, you get a cut or a scratch and they can cause an infection in a healthy person. That won't really happen in with a hospital strain, I think, well, I shouldn't say for sure, but I don't think if you had a cut or a scratch and a hospital MRSA strain infected it, you might get colonized. You probably wouldn't get really a significant infection, but the community strains you would. So it's kind of fascinating how they're offsetting that virulence and resistance.
Yeah, I was wondering because MECAR is a repressor, and if I understood correctly, oxycillin interacts with MECAR to release the repression of MECA, right?
Yeah, so I think is it MEC-I is the
or
MEC-I
MECA or MECAR,
repressor.
yeah.
MEC-OR is sitting in the membrane and it detects
okay.
protease. It detects the beta-lactam, and then when it's activated, it chews up the MEC-I, which then takes away the repression.
Okay,
but it's the same. Yeah, it's just the same thing. Yeah.
But it may not be interacting directly with antibiotics. It might be interacting with some cell wall product.
The MEC-OR,
yeah. So if we look at the Pseudomonas research on the cell wall peptidoglycan, we show what interacts with UmpR, which is response to beta-lactams, and so it's and also was very interesting in our lab. We found cross resistance when you expose to sub-inhibitory concentration. So, what happens if you just over time expose to sub-inhibitory concentration to your purine nucleotides and how it changes the final resistance pattern.
Yeah.
And so another research question.
More experiments. I know so many experiments. Aaron and Merva. Aaron is a terrific worker. There's so much to do, so much data that I have to work hard to keep up with him. So
well, we can hire an army of undergraduates. so I have a technical question. In the lab, when you do this research, when you're plating on a plate, them on a plate, can you see the colony size differences? So is there a simple selection before you go on to do the massive analysis? Is there a way to see if they have become a larger colony or smaller colony? I don't know.
I don't know. I don't believe so. Because that would be great, but I don't think there's anything visible. So a lot of the experiments, as you know, there's a lot of work in all of these experiments, the assays and kill curves, they're very labor intensive.
Yeah.
I don't believe that, the disk diffusion assays are the easiest things, to get an indication. So that's the easiest way that we have. And even they're not straightforward. When we talking about undergraduates, I had lots of undergraduates helping to screen that Nebraska library. And of course, that's a tricky business as well. For listening, because, two different people measuring susceptibility, and it can get very different results, which is kind of, so you have to be very precise and careful about how you do experiments as well.
Can I ask one last question, Dr. Puente?
Yeah, just before we wrap up.
I know I keep looking at the clock. So I just curious about how was the Nebraska library created? Is it a transposon library, and do you go back and then check recreate those mutants?
So what they did was, it's a fantastic resource for the Staphylococcus community. So they used a transposon. It's a transposon library, but they've curated it. So when you get the library, it's on 394 well plates. So you get 5 or 6 of those plates, and every non-essential gene on the chromosome has been mutated. And so as you pick them out, it's a brilliant resource. So it allows you to kind of screen, and all of the Staphylococcus groups around the world have that library, and I'm sure it's been essential for huge discoveries now. But it's all curated. So you can take each individual mutant and look at a phenotype, and then what the gene is that's being mutated. So there's an online resource to tell you what they are. So it's great. Yeah.
Very nice. Thank you. I really appreciate.
I saw a message from Merva. So
Merva.
We did a lot of the work for this.
Thank you very much, James, for a great talk. It was very insightful. And to everyone for your participation. Link to the recording will be emailed to all attendees. And the next session in the JMM seminar series will take place on Friday, July 25th. At the same time, Dr. Thuy Do from the Division of Oral Biology, School of Dentistry, Faculty of Medicine and Health of the University of Leeds will share her research in a talk titled Exploring the Low Abundant Members of the Oral Microbiome. We hope to see you all attending that talk. Thank you, James. Again, it was really, really interesting. I learned a lot about stuff, and
Yeah.
well, we'll see you next time.
Yeah.
Thank you,
Thank you
everybody
so
for your
much.
assistance. Thank you,
Yeah.
Calais.
Yeah.
Bye-bye.
And have a great weekend.
You too.